So today, I'm going to talk about DNA. DNA, or deoxyribonucleic acid, is a polymer. A polymer is a chain or sequence of smaller groups called monomers. The monomer in DNA is called a nucleotide, and there are three parts to a nucleotide: the phosphate group, the pentose sugar, and the nitrogenous base. We're going to start with the phosphate group. In the phosphate group, there is a phosphorus atom that has a double covalent bond to an oxygen atom and three single covalent bonds to three other oxygen atoms. Now, an oxygen atom wants two more electrons. That is why it will make two covalent bonds. This oxygen, however, only has one covalent bond, so it still wants another electron. So what will happen is that oxygen will absorb another electron, but that will give it a charge of -1. This oxygen, however, does have a second covalent bond, and that is to the next nucleotide in the sequence that is DNA. This oxygen also has a second covalent bond, and that is to the next part in the nucleotide, which is the pentose sugar. Now, the pentose sugar in DNA is deoxyribose, which is where the deoxyribo in deoxyribonucleic acid comes from. Now, as I mentioned, deoxyribose is a pentose sugar, and pent means 5. And the reason it has pent is because there are 5 carbons. And this is what deoxyribose looks like when it is alone. However, when it is bonded to DNA, it looks a little different. Mainly, it loses all of its OH groups. So that is gone, and that is gone. Now, each of the OH groups are replaced with covalent bonds to different things. For example, this OH group is replaced with a covalent bond to that oxygen. So that is how the phosphate group and the deoxyribose connect in a specific nucleotide. They connect via that. Now, this carbon down here also connects to a phosphate group. However, it connects to the phosphate group of the previous nucleotide. So, the previous nucleotide has the phosphate group here, and this oxygen will connect to this carbon. So that is how the nucleotides of different nucleotides connect, via that oxygen and that carbon. Now, the last covalent bond we have here is that carbon, and that connects to the third part of the nucleotide, which is the nitrogenous base. Now, there are 4 nitrogenous bases: adenine, symbolized with an A, guanine, symbolized with a G, thymine, symbolized with a T, and cytosine, symbolized with a C. Now, each of the 4 nitrogenous bases is relatively complicated to draw, and since there are 4 of them, I'm not going to draw out each one of them. Instead, I will show you a diagram I found off the internet. So, here you can see what each of the 4 nitrogenous bases look like. And you'll notice that they're labeled as either a purine or a pyrimidine. Now, you'll notice that the pyrimidines just have a hexagon, while the purines have a hexagon and a pentagon. And that is all what a purine and pyrimidine means. If a nitrogenous base is a purine, that means that it has a pentagon attached to its hexagon. That's all that it means. Now, you might also notice that each of the nitrogenous bases has a hydrogen sticking directly downwards. And this hydrogen is where it connects to the rest of the nucleotide. It connects here. So what happens is that hydrogen in the nitrogenous base is replaced with the rest of this nucleotide. So that is what a nucleotide is. And when a series of these combine together, they form the polymer DNA. So far, I've talked about the individual nucleotides. Now I'd like to talk about DNA as a whole. Now, as I mentioned, the oxygen in a phosphate group of a specific nucleotide will bond to the carbon in a deoxyribose of another nucleotide. And that is how nucleotides join together. And when you get a long chain of these, you have DNA. Kind of like the drawing I have here. We have the deoxyribose in green, the phosphate group in red, and the nitrogenous base in blue. And as you can probably tell, the deoxyribose and phosphate group form an essential backbone for the DNA. Now, DNA is a double helix, which means that there are two of these strands. And the two strands are held together via hydrogen bonding between the nitrogenous bases. Now I'm going to take a brief detour to explain what hydrogen bonding is. Hydrogen bonding occurs when a hydrogen bonds to certain other elements. So let's take, for example, water. So we've got oxygen there in red and our two hydrogens in blue. Now, the oxygen will take the electrons more often than the hydrogens. It will essentially hog them. That gives the oxygen a slightly negative charge, while the hydrogens have a slightly positive charge. This is known as being polar. Now, because opposite electrical charges attract, other polar molecules, in this case we're focusing on water, will align themselves with that polar molecule, forming a kind of chain-ish thing. And that is essentially what happens with the nitrogenous bases. The slightly positive hydrogen atoms will bond to slightly negative other atoms. They need not be oxygen. Now, another thing is that due to the chemical makeup and structure of nitrogenous bases, a specific nitrogenous base will only bond to a specific other nitrogenous base. So adenine only bonds to thymine, and guanine only bonds to cytosine. So, for example, say we have the sequence A, G, T, G. That means that the pairing sequence from the other strand of this DNA will be T, C, A, C. So as you'll notice, guanine bonds to cytosine, and thymine bonds to adenine. Now, another thing I should mention about hydrogen bonding is that it is relatively easy to break a hydrogen bond. If you add enough energy in the form of heat, you can actually break hydrogen bonds. And this is why a fever is so dangerous. If your body temperature gets too high, the DNA essentially splits apart. Not exactly something you would want. So this is essentially what DNA looks like, though. Now, because it is a double helix, that means that the two strands kind of spiral around each other, kind of like this. It's not the best drawing, but in actual DNA, the strands would spiral around each other. But still, this is essentially what the structure of DNA looks like. Alright, so far I've talked about the structure of DNA. Now I'm going to talk about what DNA is used for. And quite simply, DNA is used to make the cell. Now, what will happen is part of the DNA will split apart, like I just drew there, and the nucleotides will join up with those nitrogenous bases. The nucleotides that join up will, of course, be opposites, i.e. adenine will bond to thymine and cytosine will bond to guanine, and vice versa. And that chain that results from bonding to the nitrogenous bases of DNA is known as RNA, or more specifically, messenger RNA. Now, once the RNA has finished forming along the DNA, it will leave the nucleus, which is where the DNA is stored, and travel to what is known as a ribosome. Now, once at the ribosome, certain amino acids will be paired up with the nitrogenous bases. And that resulting amino acid chain will form what is known as a protein. Now, proteins do pretty much everything in the cell. Certain structural proteins will support the cell membrane, while enzymes, which are also proteins, will digest sugars. Really, anything in the cell that is made by the cell itself is a protein. Now, the reason that DNA itself doesn't fully split apart and go to a ribosome is that it is really, really long. Like, it is long enough to have the information for an entire cell. So it would be impractical for the entire thing to split apart and then go to a ribosome, which is why it uses RNA. Now, when a cell replicates, what happens is that a copy of each of the proteins is made, and then the entire chain of DNA splits apart. So we now have two segments of DNA, and the nucleotides will pair up to those nitrogenous bases, and they will essentially form a copy of the DNA. So now that we have a copy of the DNA and a copy of each of the proteins, the cell will then split apart. But that is what the DNA is used for, to make another cell. Alright, so that is essentially what DNA is and what it does. But I hope you enjoyed it, hope you learned something, and see ya!